Hi, this is Dr. Goldsmith again. This is the last chunk in the series that will be made in my absence. I'm going to continue talking about T cell responses. We now have activated T cells, and we want to know what their functional responses are once they see their antigens and the co-stimulation and all the other stuff that gets them over the hump to be activated. So ultimately, we want expansion of antigen-specific clones of the lymphocytes and then differentiation of what were naive T cells into effector T cells and into memory T cells. So how does this happen? And in order to understand the control of this, you have to understand a little bit about cytokines, which I have mentioned before, and I'm going to carry on here. I mentioned them before with regard to the innate immune system. And T cells in general actually secrete cytokines to a large degree, particularly the CD4 positive T cells. So just as a reminder, cytokines are a large group of proteins. They function as mediators of immunity and in inflammation, which you heard about before. You also heard about their role in innate immunity. That was already discussed, particularly with regard to macrophages. They were mentioned with regard to some other cells too, like NK cells. But with regard to adaptive immunity, which is where we're focused right now, cytokines are secreted by T cells, again, particularly the CD4 positive T cells. In the table, you've seen this before, are some general properties of cytokines. I'm not going to review them specifically, but there are some specific examples for T cells. Instead, I want to focus on some particular cytokines that are associated with T cell activation function, effector function. So once the T cells are activated, they have all their co-stimulation, they saw antigen, they had the signaling ensue, the T cells are rapidly going to secrete cytokines in response to the antigen and co-stimulators. And again, CD4 cells secrete higher concentrations and more diverse cytokines than CD8 cells do. This is probably related to their function. The CD8 cells are cytotoxic cells, so their job is to kill, and they need to be in association with other cells in order to do this, whereas the CD4 cells, they help, and they do their job of helping by secreting molecules, that is cytokines, to talk to other cells. One of the more important cytokines that are secreted by the T cells is IL-2. This is also known as T cell growth factor, and you'll see why in just a moment. It's secreted within an hour or two after activation, and it's secreted by CD4 positive T cells, and it induces survival and proliferation, hence its T cell growth factor. And if you recall on a previous image I had, IL-2 was actually shown on there and when it was expressed and secreted. The interesting thing about IL-2, in addition to being involved in this proliferation and survival, is that activation of the T cell causes an increase in its receptor expression. What do I mean by that? There's actually a little more to it than that. The IL-2 receptor is a 3-chain molecule, and naive but mature T cells, so they haven't been activated, they express only 2 signaling chains of this 3-chain receptor. This is a low affinity state. The chains are the beta and gamma chains. The naive cells do not express the final chain, which is the alpha chain, and it's the final chain that is required for high affinity binding of IL-2. That's illustrated here, where we have a naive but mature T cell. It's about to be stimulated by a dendritic cell. And I'm really focused here on the IL-2 receptor, where we have the naive cell, it's in its resting or naive state, and the IL-2 receptors in its low affinity state, meaning it has both the beta and gamma chains, but that's it. When the naive T cell interacts with the dendritic cell and everything that the T cell requires is shown appropriately, MHC plus peptide, co-stimulatory molecules like CD28 binding to B7, the activation process will allow for cytokines to be produced, including the production and secretion of IL-2. So you can see this happening now. In addition, with this activation, you get expression of the high affinity chain of the IL-2 receptor. That is the alpha chain of the receptor. So now you have a high affinity complex of the IL-2 receptor that includes the alpha, beta, and gamma chains. They bind IL-2 with greater affinity, and IL-2 in turn does its job much better now, which is to induce T cell proliferation. So that's really very, very cool. This is just an image to give you an idea of some of the cytokines. We're not going to talk about all of them in this next portion of the video, but IL-2 is listed on here, and you'll actually hear a lot about other cytokines like IL-4 and IL-5 or IL-17 as we continue through the course of the semester. You have heard about gamma interferon with regard to activating macrophages. So, lest you think I'm making you memorize yet another molecule, I actually do so for a very good reason, and that is IL-2 is used in therapeutic agents. There are huge applications for IL-2. So there are multiple therapeutic agents that target IL-2, again, T cell growth factor. Some are monoclonal antibodies that antagonize the IL-2 receptor. What do you think this would do immunologically? Some therapeutics agents are just infusions of IL-2. What do you think this would do immunologically? And one is a fusion protein where diphtheria toxin, so a toxin, is fused to IL-2, and the fusion protein will bind the IL-2 receptor alpha chain. What do you think this will do immunologically? I'm actually not going to answer these. I want you to figure these out or find information about them, and I promise you I will ask you at least one question on the quiz about the specific application of these therapeutic agents. So we now have T cells that are clonally expanding. It occurs within 1 to 2 days. You get a huge expansion. After activation, you get a huge expansion of antigen-specific clones. This was just described, particularly with regard to IL-2. And essentially, this is great because it keeps pace with microbial cell division. So the T cells are trying to keep up with the microbes and provides a large pool of antigen-specific T cells for effector function, so they can have their effects. There are some really unique features about the expansion. The expansion is specific, meaning other cells, bystander cells that haven't recognized antigen, they will not be proliferating. So there's no detectable increase in bystander cells or non-specific cells. Only those cells that have recognized antigen and gotten co-stimulation. The majority of the clones that are proliferating are specific only for a few of the antigens in the microbe, the antigens that are recognized by the T cell receptors, and the antigen in association with MHC. So you see a few different clones expanding, but certainly not all T cells will be responding. Another interesting thing is that the expansion of the CD8 positive cells, so proliferation and differentiation into their effector forms, is much greater for CD8 cells than it is for CD4 positive cells. Again, this probably is not a big surprise since CD8 positive cells need to move and get out to a place to associate with a cell to have their effects, whereas CD4 positive cells, they're helper cells, they actually secrete cytokines, and the cytokines can move. So you don't need as many of them to secrete. So it's not surprising that the CD8 positive cells expand to a greater degree. The expansion, just to give you numbers, I'm not going to hold you to this. It's amazing, actually. Before infection, the frequency of a CD8 positive cells for any specific antigen is on the order of 0.001 to 0.0001% of all the lymphocytes in circulation. But at the peak of some viral infections, say about a week after the infection, the antigen-specific lymphocytes can comprise up to 10 to 20% of all the lymphocytes in circulation. That's a 100,000-fold increase. and the cells are doubling on the order of every 6 hours. It's incredible, actually, how much the cells proliferate. Again, very specific proliferation, only those that recognize the antigen. Differentiation into effector cells occurs later in the proliferation phase, about 3 to 4 days of rapid growth and differentiation occurring, and as should not be a surprise that there will be changes in gene expression and therefore protein expression as a result of this change into an effector cell. You're changing the phenotype of the cell. The effector cells are going to make the molecules that are required for specific functions, whether that's a helper function and the cytokines they'll be producing, or whether it's cytotoxic T cells and the effects that they'll be producing with regard to their granule contents and how they kill effector cells. The effector cells have some distinguishing characteristics. Again, they're changing their phenotype. In contrast to naive cells, the effector cells no longer need co-stimulation to be activated. Encounter with their specific antigen that they recognize is enough to activate them. So this is very much in contrast to a naive cell that needs that co-stimulation. In addition, there'll be changes in cell adhesion molecules and receptor expressions. Again, not a surprise. This is going to be so that the cells can move to site of infection if necessary, and I'm actually going to come back to that a little later on. So I have a question for you, and that is, which of the following is or are effector cells? I'll let you think about this a little while. All CD4 positive T cells, all CD8 positive T cells, all CD4 positive and CD8 positive T cells, or CD8 positive cytotoxic T cells. And the answer is the CD8 positive cytotoxic T cells. The first three choices, they could be in their naive state, just because they're positive for CD4 or CD8, doesn't mean that they have been activated, proliferated, and differentiated into effector cells. So only 4 is correct. And speaking about effector cells, what are these effector cells? Well, remember, a naive T cell is either going to bear a CD8 or a CD4 co-receptor on its surface. The CD8 positive or CD8 bearing cells all are going to differentiate into cytotoxic T cells. That's the effector cell, also known as cytotoxic lymphocytes, cytolytic lymphocytes, or CTLs, and their job is to kill target cells, as I've said a few times. The CD4 bearing cells differentiate into multiple effector cells subsets. This would include Th1 or T helper subset 1, Th2 cells, T helper subset 2, there are Th17 cells, there are regulatory T cells. All of these have CD4 on their cell surface, but they have different functions, which we'll talk about in greater detail later on. I do want to mention something with regard to CD4 positive helper cells. So we're talking about effector cells here. So they are helper cells, they are effector cells. When they respond to antigen, one of the things that they do is produce surface molecules and cytokines that activate other cells. In other words, they're going to help other cells like phagocytes and B cells. And one of the molecules that is very helpful for this is known as CD40 ligand, or CD40L. It's a surface molecule that's found on the T cell. And the CD40 ligand gene is transcribed in response to antigen recognition and co-stimulation, and so it's expressed after activation into an effector helper cell. The CD40 ligand on this effector helper cell is going to bind CD40, and CD40 happens to be expressed on things like macrophages and B cells. It's also expressed on dendritic cells. The binding of CD40 to CD40 ligand on the effector T cell activates those CD4 positive cells to do their job secreting cytokines. So this is another nice way that we get responses from the helper cells. So this is illustrated here, where we see in the top panel, a CD4 positive cell helping a macrophage in green, and in the bottom panel, a CD4 positive cell helping a B cell. And really the focus here is you have an activated effector cell, the CD4 positive helper cell, and it's going to do its job, which is to help a macrophage. So here we have it helping a macrophage. The macrophage is showing antigen on MHC. The MHC would be class 2, of course. And the focus here is CD40, which is found on the macrophage, binds CD40 ligand on the effector helper T cell, and that induces signaling that results in cytokines being produced that can then help the macrophage do its job better, which is, of course, to kill phagocytosed microbes. The same thing can be said for a CD4 positive helper cell helping a B cell. Again, the focus is on the interaction between the two here. The B cell is showing antigen on MHC that was processed, MHC class 2, to the T cell receptor on the helper effector T cell. The B cell has CD40 expressed on its surface, while the T cell has CD40 ligand on its surface. These two things bind. This helps send signals within that helper T cell to secrete cytokines, which then allows the B cell to do its job, which is, of course, secreting antibodies, and it helps to enhance the ability of the B cell to differentiate and do that. We'll come back to these concepts again when I talk about more specifically about the effects of the helper cells and the cytotoxic cells, all the effects of the effector T cells. A final thing to think about with regard to activation and then differentiation into effector cells, the T cells that are activated, some of them are actually going to develop into memory T cells. So a portion of the antigen-activated T cells are going to differentiate into long-lived memory cells. These memory cells survive even after the infection has been eradicated. They're found centrally in lymphoid organs. They're found in the periphery in mucosal tissues. So we have both central memory cells and we have effector memory cells. The effector cells would be found out in the periphery. The central cells would tend to be found in the lymphoid organs themselves. Both of these are able to respond rapidly, clonal expansion upon exposure to their specific antigen, which is beautiful. And in order to do so, they require cytokine signals to survive so that they are able to respond. And that includes things like IL-7, which was mentioned before with regard to the appropriate environment for the development of our lymphocytes. This was produced by stromal cells and was described with maturation of lymphocytes. Again, there are two types, the central and the effector memory cells. The central tend to be found in the lymphoid organs, and the effector memory cells tend to be found out in the periphery, like in mucosal tissues, for example. The unfortunate thing about memory T cells is that the factors that determine if the progeny of an antigen-stimulated lymphocyte, in this case the T cell, is going to differentiate into an effector or a memory T cell, we don't know. Hence the question marks, which is a big, big bummer. All right, the last thing I want to talk about is T cells are part of cell-mediated immunity, and as such, they move. They move a lot more than the B cells do. And so how do we get migration of the T lymphocytes in cell-mediated immune reactions? So once the T cell has been activated by binding of antigen and co-stimulation, what are the responses? How do they ultimately combat microbes, and how do they get to the locations where they need to do that? So this should sound familiar because I talked about these general concepts with regard to neutrophils extravasating. And in a similar way, we have T lymphocytes with different phenotypes depending upon whether they're naive or activated or effector cells. They have different characteristics, and these different characteristics allow them to move to different places. So a naive T lymphocyte will enter the lymph nodes, it's in circulation, and there are chemokines and adhesion molecules that help with this process. There are adhesion molecules on the naive T cell, like L-selectin. There's an integrin, LFA1, which you've seen before. There's a chemokine receptor, CCR7. You've actually seen this before with regard to dendritic cells getting to lymph nodes. So these molecules are found on naive T cells in circulation, and it provides them the ability to go to the lymph nodes. You have to have corresponding molecules on what are known as high endothelial venules within the lymph nodes. These are essentially blood vessels that lead into the lymph nodes. So you'll have ligands for the selectins that we see on the T cells, L-selectin ligand, for example. And you'll have ligands for the integrins that we find on the naive T cells. In this case, the high endothelial venule will have ICAM1, or intracellular adhesion molecule 1, which binds LFA1. Again, you've seen a couple of these before, actually. And of course, there are going to be chemokines that direct the cells to the lymph nodes and will be involved in activating the integrins for improved binding or increased affinity. Now, cells go through circulation. They're able to perhaps interact with dendritic cells. Maybe they'll see an antigen they like presented by that dendritic cell. Maybe they won't. If they happen to see an antigen they like, they are able to migrate to the site of infection, and this is based upon the change in phenotype that results from signaling that occurs from antigen recognition and from co-stimulation. So, once you have an effector T cell, migration to the sites of infection depends upon expression of adhesion molecules and chemokine receptors on the T cells, expression of and display of ligands on the endothelium at the site of the infection, and response to chemokines produced at the site. Again, this is no different than what you saw with neutrophils, just slightly different molecules. And the cool thing is, when the naive cell differentiates into an effector cell, the change in phenotype is accompanied by changes in the adhesion molecules and the chemokine receptors that allow that activated, now differentiated effector cell to move to the site of infection. So, what are some of these molecules to allow that differentiated lymphocyte to enter the tissues? Well, we have adhesion molecules on activated T cells, not a surprise. That includes ligands for selectins like E and P selectin ligands. It includes integrins again, LFA1 happens to be one of them, VLA4 is another one, it's a new one. And then, of course, you need corresponding molecules on the endothelium at the site of the infection. E and P selectin is there, you've heard this before. Ligands for integrins like ICAM1 again. VCAM is what binds VLA4. And so we have the appropriate molecules interacting at the site of the infection. Chemokines, again, are going to direct the cells to the sites of infection and activate the integrins to improve their binding. Again, this is nothing new. And it's actually really nice because it allows the T cells to move to the site of infection. And in addition, the T cells, I didn't mention it on here, but the T cells would reduce the expression of molecules that would keep it in the lymph node. So again, more encouragement to leave that lymph node. As long as it's an effector cell, leave the lymph node and go to the site of the infection. So this is illustrated here. We have the T cell, a naive T cell represented in red, a couple of them here, an activated T cell in green. They move through the circulation. They go through the high endothelial venule into the lymph node following their chemokine trail thanks to the CCR7 receptor on their surface. The naive T cell is going to interact with a dendritic cell if it sees its antigen plus MHC plus all the co-stimulatory molecules. It's going to become an effector cell. I'm going to point out that the molecules that were just described in text, some of them are shown here, to really get you thinking about what is expressed on the high endothelial venule in the lymph node and what is expressed on the T lymphocyte in its naive form. So you'll notice that L-selectin is expressed on the T lymphocyte. integrin LFA1 is expressed on the lymphocyte, and you have the corresponding molecules on the high endothelial venule, that is the blood vessel entering the lymph node, so that L-selectin will bind to ICAM. Excuse me, to L-selectin ligand, my mistake. And then LFA1 will bind to ICAM1, allowing that T cell to move into the lymph node following its chemokine trail. Once that T cell is activated, you get expression of different molecules on its surface, including chemokine receptors. It will follow a different chemokine trail. So I'm going to focus here first on some of the molecules that were mentioned in text before. We now have different type of chemokine receptor being expressed, so the T cell that is an effector cell can follow the chemokine trail. We have E and P selectin ligands being expressed, which are going to bind to E and P selectin on the endothelium at the site of the infection. We have integrins expressed, again, LFA1 or VLA4 expressed on the effector T cell, and these will bind to their ligands, ICAM1 or VCAM1. What was not described in text before is something really interesting. You know, the T cells are going to follow a chemokine trail to get to the tissues. But there's a phospholipid known as sphingosine 1-phosphate, or S1P, sphingosine 1-phosphate, that is expressed on effector cells. It actually plays a role in the exit of T cells from the lymph nodes by binding its receptor called sphingosine 1-phosphate receptor, type 1. And that's shown here. There's the receptor. So what happens here? It turns out that levels of sphingosine 1-phosphate are actually higher in the blood and lymph, so in circulation, and bind many of the sphingosine 1P or phosphate receptors on the circulating T cells. And that actually keeps the expression of the receptors on the T cells low. But when the T cell enters the lymph nodes, the sphingosine 1-phosphate concentrations are lower there, and that typical response of a cell, and this happens for the T cell here, is to upregulate or increase expression of the sphingosine 1-phosphate receptor. So it does that in the lymph node when the T cell arrives. If the T cell doesn't recognize antigen, the T cell will follow that sphingosine 1-phosphate gradient. It's actually higher in the lymph and the blood, as noted earlier. It will follow it out into the efferent lymph vessels, so following the higher concentration gradient. But if antigen is recognized in the lymph node, so something like this is happening, then the expression of the sphingosine 1-phosphate receptor on the T cell is actually going to be suppressed for a few days. And that means that the T cell is going to stay in the lymph node and allow it to clonally expand and proliferate and differentiate into an effector cell. Once that clonal expansion and differentiation is over, the sphingosine 1-phosphate receptor is increased. Its expression is increased. And again, you will get the effector cell following that trail of sphingosine 1-phosphate in the blood and lymph. So it's calling to it to move out into the blood and lymph. At the same time, expression of the CCR7 receptor would decrease, and it's the CCR7 receptor that calls the T cell, the naive T cell, to the T cell zone of the lymph organs. So that's really a cool thing, and these are things that can be exploited and have been exploited therapeutically. So it's really very cool. This is a table for those of you who like things presented this way, which focuses on the receptors and their ligands. I will point out that there is a differentiation between a naive T cell, or naive T cells that are shown in the top of the table, and activated effector and memory cells that are shown on the bottom. I'm not going to read through this. I think you can do it yourself. Many of these molecules, in fact all of them, have been mentioned to some degree or other. This is a really nice summary that I think will prove helpful to you. And one final thing I would like to point out for you with regard to the movement of the T cells, it's independent of the antigen recognition. Really all movement depends on is the effector status of the T cell. So if a cell is an effector cell, it gets to the sites of infection via its adhesion molecules and chemokines. Antigen receptor being displayed is not important. It will be displayed, but it doesn't matter. What matters is the effector cell status. In other words, that the phenotype has changed, and so you're getting expression of different adhesion molecules and different chemokines because you now have an effector cell. So any effector T cell would enter a site of infection. It's non-selective. What matters once it gets there is effector cells that recognize the antigen at that site will stay at the site. If they don't recognize the antigen at that site, they'll keep moving on until they find a site where they do, as long as they're an effector cell. So binding of integrins to the extracellular matrix in the tissue keeps the cell there long enough to bind antigen and perhaps recognize it. So again, it doesn't matter, the movement doesn't matter whether what the antigen is at any particular site to get it there. What matters is whether it's an effector T cell or not. So I have a select all that apply question for you. Leukocyte adhesion deficiency is a genetic disorder in which LFA1 is not properly expressed. What will be the outcome of this disease? It will result in, and there's more than one answer, but not all answers are correct. It will cause reduced lymphocyte adhesion. It will cause increased lymphocyte activation. It will cause improved T lymphocyte responses or decreased response to intracellular expression. So LFA1 not being expressed appropriately is going to cause reduced T lymphocyte adhesion and decreased response to intracellular infection. Because LFA1 is one of those proteins necessary for cell adhesion, if you reduce how much is expressed, it's not properly expressed, the cell is not going to move appropriately, so cell adhesion is going to be reduced, and that means its response to infection is going to be decreased. It has nothing to do with activation. It's certainly not going to help improve T cell responses. So hopefully that makes sense to you. And my last thing before I wrap up is the decline of the immune response. Of course, we don't want our effector T cells out there working all the time. We want to return to steady state or homeostasis. And this occurs over time when survival and maintenance signals for the T cells are removed. So as we clean up a microbial infection, antigen will be removed, co-stimulatory signals from, say, CD8 binding to B7, they'll be removed. Cytokines like T cell growth factor, IL-2 will be removed because we no longer have antigen around to activate the system. The cells that were responding will do their jobs until they no longer see antigen, and then they'll die via apoptosis. Typically, a response subsides within 1 or 2 weeks after the infection is eradicated. But of course, you will have a sign of the infection remaining, and that is the pool of your memory cells. That is all I have. Thank you very much for listening. I hope this was clear as a bell. I will be back in the office on Monday. If you have questions, certainly you can email me, and I will be doing my best to get to those questions upon my return. Thank you very much.